Comparative genomics compares, as it suggests, compares the DNA data, sequence data, from different species to try to help understand how genomes function and how they evolve. So by looking for similarities between species and taking what we know from one species and then applying it and seeing if it's also true in the other species. So it's a cross-species comparison of genomic information. These are some of the completely sequenced genomes in the prokaryotic group. You have the archaea here and the eubacteria. Here's E. coli down here. And what you can see from the sequence, even though there's differences, the size of the genomes really doesn't exceed 10 million base pairs. So this one being pretty large. E. coli has a 4.6 million base pair size of a genome. And to keep this in perspective, an average human chromosome, one chromosome from human, has about 100 million base pairs on it. So the genome size, at 4 million or 9 million base pairs is, quite small. The number of genes on any of these genomes, doesn't exceed, I guess, 8,000. Looks like it's the biggest number here. And E. coli has about 4,000. So pretty, small number of genes. But I guess what's interesting is that a human chromosome, even though it's 100 million base pairs long, typically only has about 1,000 genes. So even though the chromosomes individually are much longer than, say, a bacterial chromosome, they contain actually, less density of genes. So more junk sequence in a eukaryotic chromosome. So the prokaryotic chromosomes are relatively small and have not a ton of genes, but the genes are very compact. There's not, junk sequence between the genes. Moving into the eukaryotic genomes, you see that the genome size starts at 12 million and goes up from there. Humans have 3.2, billion base pairs. And, but the number of genes doesn't increase in a proportional way. So you have about, 24,000 genes. We talked about in mouse, about the same. And yet the genome size is enormous. So, we have 6 or 8 times the number of genes of a bacterial cell, but, 3,000 or 1,500 times the size of, number of base pairs. So what that means is by increasing the size of the genome, you're not getting a proportional increase in the number of genes, you're getting a lot of junk sequence. Now looking at the results of the E. coli genome project, they've been able to computer predict what genes are there based on what genes we've already discovered and what is extremely similar to genes we've already discovered, and then classified them into some broad categories here. And what you'll notice is there's a pretty large category, number 2, which is function unknown. So a pretty big chunk of the pie, I'd say a third of the pie, looks like genes, looks like bacterial genes, but we don't know. They're things we've not encountered or discovered or described before. So that's interesting that there's potentially something happening there genetically that we're just not even aware of. Some other things, when you compare genomes, percentage of genome consisting of repetitive elements, especially transposon-derived elements, humans have 44% of their genome consists of these repeated elements. So ALU being one of them, but others as well. So a lot of junk, repetitive type sequences in the human genome. You don't see an equal amount of that in some of these other model organisms. Drosophila only has 3.1% composed of these repetitive transposable element type sequences. So now we look at the layout of the human genes. We said there's about 24,000 human genes, or genes in the human genome. And when you look at the breakdown, similar to what we looked at in E. coli, when they look at the sequence, they have a computer that predicts where the genes are. The computer looks for promoter sequences, terminator sequences, codons, intron and exon splice sites, those types of things. Then it identifies what it thinks will be a gene. And of course sometimes the computer is probably wrong, but depending on the stringency of the algorithm, we can come up with a reasonable estimate. And when they look at when you kind of make the same pie chart of the genes that the computer finds, cross-reference to the genes that we've already identified or things that are very similar to what we've already identified, again, you end up with this huge piece of the pie. In this case, it's number 17, and 17 is molecular function unknown. And I would say it's more than 30% in the human pie. It's more like 40%. So what this means is that more than 40% of the genes in the human genome have no previously known function. And what that suggests to us is that there's a group of genes that's doing something and we're completely unaware of it. There's some big part of cell biology that we're missing. Or there could be a lot of small things happening and we're missing all of them. But there's, we think that we know a lot about cell biology, and we do, but there's something big going on in genetics in the cell. There are genes in there doing something and we just are completely missing it. Now, one interesting story in terms of comparative genomics has been done by studying the genomes of the different Drosophila species. And you can see here the Drosophila species, of course, Drosophila melanogaster, you're aware of, but all these other species as well. And by the completed sequencing of the genomes, it was called the Drosophila Genome Consortium, they were able to study and compare and this is just a summary, obviously, of how many genes the computers find and size of the genome, and then by various algorithms you can put the species into groups of evolutionary relationships. So along the x-axis here would be time. So if you're not familiar, this is an evolutionary tree, and then as you go back in time, it shows the branches coming together. That shows where the common ancestors would occur between these groups. And so, for example, these two here, Drosophila simulans and Drosophila secalio, I'm not even familiar with that name, but those are species. They have a common ancestor here. And then a little further back in time, they share a common ancestor with Drosophila melanogaster, which is the fruit fly you're familiar with. So by doing these comparative analyses, they can put together, based on the data, these relationships. And of course, new data might shift things a bit, but this allows you to study, really detailed evolutionary relationships of organisms. This just being one example.